Greetings, friends, fellow Earthlings, and fans of exploration. Welcome to Ask An Astrobiologist, the show that celebrates the science and celebrates the scientists involved in our quest to understand the nature of life. Astrobiology. Where does life come from? How does it change and evolve? Where is it located in our universe? The questions we all want to know. I am your host, Dr. Graham, the cosmobiologist Lau. And as always, we're brought to you by seganet.org and the NASA Astrobiology program. As always, we want to thank all of you as well for tuning in, asking us questions during the live stream chat, or if you're watching this later, jumping over to the YouTube channel and adding your questions in. I do my best. Several of us come through and look at those questions, try to answer them when we can. I'm so excited for this week. Now, you might notice I'm in a slightly different environment. I'm actually recording in Savannah, Georgia, for this month's episode. But I'm so excited to have our guest on the show this month. Dr. Andrew Schuerger is joining us. He's a scientist at the Department of Plant Pathology at the University of Florida. Dr. Schuerger worked for 18 years at the Land, a hydroponic research and education facility at Disney's Epcot Center, as well as at NASA's Kennedy Space Center. His research has gone in many different directions over the years. It includes studying the effects of Martian conditions on the survival, growth, and adaptation of microorganisms, as well as investigating things like methane on Mars as a potential biosignature. And so I'm so excited to welcome Dr. Schuerger, and I hope you will as well. Dr. Schuerger, thank you very much for joining us for Ask an Astrobiologist.
Oh, hey, it's my pleasure. I'm glad to be here.
Like I said, I'm so happy that you're joining us. Looking through your bio, you've done so many different things. You earned your bachelor's degree, your master's degree at the University of Arizona. Then you worked during your PhD. You earned that in 1991 at the University of Florida. Working in this realm of microbiology, microbial ecology, and then plant pathology, I'm wondering for our audience, could you explain what really drove you towards these realms of like microbiology and plant pathology?
Well, the biggest reason I got into plant pathology is I immediately had the sense that it was like being a detective, because you have a series of problems with a plant or a crop. There are symptoms or signs. You have to go into it and kind of use all of your knowledge from every course you've taken, from every bit of experience that you've been able to garner, and then figure out what the problem is. And then that's first level of detective work. And then the next level is to figure out how to control that disease. So I just thought it was a very challenging, innovative discipline, and I've been very pleased with a career in plant pathology.
That's wonderful. And of course, you're now at University of Florida, but even while working on your PhD, you were working at the Land at Epcot Center, kind of in this realm of hydroponic research and education, looking in these management systems. I'm wondering, how did you kind of evolve your career into that realm of like looking in closed loop systems and hydroponic systems and kind of using these as a way to understand plant pathology?
Well, that's a very interesting question, because when before I applied for the position at Epcot Center at the Land Pavilion that I eventually got, I was very interested in going into nature cinematography. But my department chair in plant pathology at the University of Arizona tracked me down in the hall carrying a sheet of paper, and he said, "This job's for you." And it was the announcement of working in a complex hydroponic facility, up to 40, 50 crops, agronomic and horticultural crops. And I immediately, right when that department chair showed me that piece of paper, made the connection that is sort of like a ground test bed for trying to learn how to grow plants in outer space. So that was the reason I applied, was that NASA was here in Florida, Kennedy Space Center was here in Florida, Epcot was here in Florida, and this facility growing plants hydroponically was like a ground test bed for doing it in space. So I said, I have to try. And I was lucky enough to get it. And it was a real rewarding experience working for Walt Disney World.
Oh, that's really cool. I do have to ask, since I really love this realm, not just of astrobotany, but also the future of agriculture. What your view is kind of on the future of like things like vertical farms and indoor greenhouses. Do you think we kind of need to go that direction for not just being sustainable for space exploration, but for building a future for humans as well here on Earth?
Well, that's a good question too, because vertical farming has a real important role to play in urban areas where you're growing high cash value crops, strawberries, lettuce, dwarf tomatoes, things of that nature, such that you have a very short distance from production facility to sales facility. But it's generally economical only if it's a high cash value crop like the ones I just mentioned. For bulk growth of plants like wheat, potatoes, some of the staples, that's probably going to remain for the foreseeable future as a field agriculture technology. So we really need both. We need solid field agriculture with good control of soils and pests and proper nutritional management of the crops, ecologically sound growing, as well as vertical farmings for very targeted communities. But the link here is vertical farming is a very good proxy for learning how to grow plants in really small spaces under really tight light and temperature controls that would extrapolate to growing on the surface of Mars or in a habitat on the moon.
Oh, that's wonderful. Yeah, and there's so much that we need to know for better growing food in our spacecraft. We've now grown plants on the ISS that astronauts have eaten, homegrown food on the ISS, but it feels like that realm is still kind of developing. What do you think are some of the key things we need to know right now for the next maybe decade or two, and when it comes to growing plants on a space station, moon, or on Mars?
Right. Well, the number one thing is that I think a lot of your viewers are probably seeing a lot of very complex illustrations, artist renderings of a moon base with a very complex agricultural system inside, or a Mars base with something similar. That's the target, that's the goal 10, 20 years down the road. Right now, though, we're really focused, we being the community of astrobiology and plant biology in space, we're really focused on trying to optimize plant growth in the microgravity environment on space station. A number of plant growing systems have been developed, they've flown, and to me, as a plant pathologist, one of the most intriguing issues that does not have a lot of research is how do the microorganisms present in, let's say, the space station, or eventually those same microorganisms might be in a bioregenerative life support system on the moon or Mars, how do those microbes interact with the plants? And is the space environment so stressful to the plants that host resistance that we normally expect from a plant, would that break down because of the stresses in microgravity? And that's an area that I'm actively doing research in right now, and it's quite enjoyable and quite challenging.
That's fantastic. And I do want to talk then a bit about how things will survive on Mars. Earth life, we are protected here on our planet by this beautiful magnetic field that's generated by the dynamo of the outer core of our world. And I think some people forget sometimes that, astronauts and other terrestrial organisms going to Mars, they're going to face a different radiation environment at the surface of Mars, as well as things like
Right.
the very low pressure, the much lower temperature. It's an average of negative 90 Fahrenheit at the surface of Mars, even though that does drastically change around the world. But first, for our audience who are watching live, we have a quick poll to drop in the YouTube chat for those of you who are watching right now. We have a question about radiation and how it affects not just humans, but some of the extremophiles of our world as well. So we humans, we get sick with radiation when we get a dose that's actually pretty close to the dosage we would get on the surface of Mars, around 0.3 grays or so. A gray is
Right.
1 joule per kilogram. That's the absorbed dose of radiation. And so our question for you in the YouTube chat, and we'll come back to this in a little bit, is how much radiation do you think the extremophile Deinococcus radiodurans can handle? And you'll see the answers popping up right now in your chat. Andrew and I can't see those answers, but we'll come back to that in a little bit.
Okay.
Andrew, from you, I just want to hear, what your research has been, kind of looking in this realm of survival and growth and the adaptation organisms have had to use to survive in Martian conditions.
Right. So I sent a slide with the Mars chamber on it. If you could just pop that up briefly. I've designed and built a Mars simulation chamber here in my lab, have been working with it almost 20 years, and it is able to recreate 5 conditions on the surface of the current day Mars. Low pressure, which is around 7 millibars. I'm in Florida near the sea level. Near sea level, so I'm breathing right now 1,015 millibars, but the surface of Mars is actually 7 to 10 millibars, so it's less than 1% of sea level pressure on Earth. So that's the first factor. Second is UV irradiation, very strong ultraviolet irradiation environment on Mars, because there's literally no ozone layer in the Martian atmosphere, which allows a lot of the biocidal UV to hit the surface. Number 3 is temperature, gas compositions, number 4. And then the Mars chamber also has a way of simulating either dust-free conditions on Mars or global dust storm conditions and everything in between. So what I've done, if you can then switch to me for a close-up, I have these little microbial sample holders. This is just one example. And this a little piece of aluminum in here is what's called a coupon. And upon each of these coupons, I would put a different bacterium. And these are examples just with a felt tip pen just so they'll show up. But if you could think, the blue coupons are one species of bacterium, and then the red is a different species. I would place this inside the Mars chamber, allow the ultraviolet irradiation to come down, strike those samples, and it's just looking for survival. How well can the bacteria and fungi survive just exposure to that solar ultraviolet irradiation? And it turns out that if the bacteria and fungi are exposed out on a flat surface, or even if they're kind of shielded a little bit, enough solar UV irradiation bounces around and strikes them that populations as high as 2 or 3 million spores per square centimeter can be killed off within 3 or 4 hours on the surface of the equator of Mars. Just within that first sol. And if you didn't know, a day on Mars is called a sol. So in summary, from a survival standpoint, exposing bacteria and fungi just to the low pressure environment, maybe we lose about 20% of a population over a few sols. If they're hit with solar UV irradiation, they're killed off very, very quickly. But those are only 2 factors. There's another 18 to 20 factors that have been discussed for the surface of Mars. High salts in the soil, volatile oxidants in the soil and atmosphere, solar UV radiation, solar particle events from the sun that might strike the planet. And I won't go on, but there's at least 20 biocidal factors. All of them working synergistically to reduce the population of spores, cells of both bacteria and fungi on these spacecraft.
Wow, that's rather incredible. And so I will come back to our poll for our audience then with that too, since you just said, some of these spores will thrive. Like, you have this slow kind of process of some things dying off. When it comes to life on Earth, there are different kind of ways for organisms to adapt to the radiation environment. For instance, in different places, E. coli, for instance, can actually take a much larger dose than humans can. I think the last I read was something like 200 to 700 grays was kind of an average decent dose that E. coli could still thrive in, which is higher than the Martian surface. For our poll for Deinococcus radiodurans, which, by the way, still holds the record that we know of for a radiation dose, it looks like most of our respondents said either 100 grays or 1,000 grays. But indeed, the answer was 5,000 plus. They can take a dose well over 5,000 times what would get a human sick and still thrive. Even tardigrades have been put into 4,000 gray dose environments and have survived. And so this is, those are very high radiation environments. We put Deinococcus radiodurans into the cooling fluid from nuclear radioactive plants for energy, and they've survived inside of that cooling fluid. And so, these things can take a pretty hard blast, but then you start adding in all these other conditions, just like Dr. Schuerger mentioned, with the low pressure and then salts in the soil, different gas compositions, dust. And those are the things that we need to study together in a systems approach. And so it's really cool your lab is doing that with this Mars chamber. Have there been any really kind of exciting or surprising results maybe from that research with the Mars chamber?
Well, I can't go and answer that question until I make one comment on Deinococcus radiodurans. I was sort of shuffling around there just a moment ago because I literally downloaded a paper this morning that talked about Deinococcus radiodurans surviving exposure up to 120 kilograms. Now, a kilogray is 1,000 grays, so that would be 120,000 grays. The reason why humans have sort of like a very severe reaction to even extremely low dosage of radiation is that radiation is exposing billions and billions of cells in our body, whereas a bacterium is a single cell sitting out on a surface. So you have to get the intensity dramatically higher to get that same lethality. It's just a body mass issue. That's the difference between human survival and a bacterium surviving. So I digress. What was the question?
Well, that was a very good point. So we did see that paper as well in planning for this episode, and it was just so shocking that the radiation dose was that high, that we just did 5,000 plus, until there's some more results to support that conclusion. But it is a rather incredible conclusion. But I think, my big question, using this Mars chamber, really having kind of the systems approach now to multiple different scenarios, these 5 different conditions that we may experience on Mars.
Right.
Have you had any really, surprising or drastic results that kind of changed how you're thinking about life on Mars?
Yes. About 10, 12 years ago, I started research moving from survival, which is, in my view, survival is a term that I would use for dormant or inactive cells or spores persisting through time. They don't have to replicate. They're not taking in nutrients, metabolizing, and dividing. So survival could just be a dormant cell lasting a long time. I did a lot of work with survival in the Mars chamber and under UV irradiation and vacuum and temperature extremes. And then I started getting interested in bacterial growth, metabolism and growth. And so I was interested in determining at 7 millibars of pressure, at a 0 centigrade, and with enough liquid water present to keep cells hydrated, could any terrestrial bacteria actually metabolize and grow under these Martian surface conditions? And I did experiment after experiment, bacterium after bacterium, and I literally was just ready to say, okay, I'm tired to write a paper that says nothing grew under Martian conditions. And the very last bacterium I tested actually grew under the low pressure, low temperature, and gas composition in the assays. So it was a really pleasant surprise. And once you establish a protocol that confirmed that this bacterium's name, by the way, is Serratia liquefaciens. It's a non-spore forming bacteria found in spacecraft and on surfaces of hardware, mainly hardware associated with human activity, like on the space station or Apollo spacecraft, Skylab, things like that. But in any event, once I have the basic protocol that I know something will grow under Martian conditions, then we could start going through large numbers of bacteria a lot faster. And to date, we've identified approximately 30 bacteria, but no fungi so far, that can grow, metabolize and grow under low pressure, hydrated conditions similar to the surface of Mars. So that just with one bacterium, one last test, changed my whole perspective on what might be possible on the surface of Mars.
Wow. Yeah. Instead of being eureka, it was a I didn't think that would happen kind of moment. I love that so much. And that kind of also then is important for my next topic I kind of want to discuss here is the idea of forward contamination. There's a whole department, an office at NASA that does planetary protection, figuring out, how do we, just not only avoid contaminating Earth with possible things from elsewhere, but how do we avoid taking Earth life and contaminating other places? And, no matter how much we try to clean, our cleanest clean rooms are still going to have some biology. We're still going to send some things with our rovers. And when we send humans, it gets even more crazy because, we humans have a microbiome and we're shedding off organisms all the time, not just our own cells, but other organisms with us. And so, we'll have this moment when we get to Mars, when humans start exploring, especially where, we are, putting out earthly organisms on the surface of Mars. And I know you've done some work in that, looking at how we're going to have some issues with forward contamination. I wonder if you can speak to that issue with how we might, try to avoid, dumping a bunch of organisms on Mars, or can we even avoid that at all?
Well, we have to step back a little bit before we go to that specific question. Look at any image of a rover, like the Perseverance rover right now, or Curiosity, and you see this structure, these fairly large structures with all these little nooks and crannies in it. But everybody's got to kind of keep in mind that even if there are bacteria and maybe a few fungi and a few of the prokaryotes called archaea, if those are all present on the surface, that surface is being immediately bathed on as soon as it lands in an extremely strong ultraviolet radiation environment. And photons of light are incredibly small. Scientific community doesn't even have a consensus on how big they are. The best estimate I have is 10 to the minus 18 centimeters as the upper limit of how big they might be. It's believed to be much smaller. So these incredibly tiny little energy packets can get into almost every pit, every crack, bounce around, bounce off of one surface to get into a shaded area. And I've been doing modeling, several papers out, working with other colleagues to try to push the modeling. And the UV environment alone is so strong on the surface of Mars. Most of these surfaces of spacecraft and rovers are being sterilized within a few sols for the upper deck and maybe a few months for the stuff that's underneath on the belly of the spacecraft. So it's not, the bacteria are not lasting for generations. It's very likely that the microbiota on these rovers is being inactivated very quickly. Okay, so we pause there. Now let's look at human missions. Human missions, yes, humans will have a much richer biome that they will bring with them, but that biome is contained within a habitat. And when they go out and do an EVA, that microbiome is contained within their spacesuits. So when an astronaut, just think of this as just conceptually, they may have contamination on the outside of their spacesuit. They walk out into that incredibly rich, intense UV environment on Mars. We're not going to do EVAs at night because of temperature issues and lighting issues, so they'll almost always be done in the daytime. And that the data from multiple labs supports the conclusion that most of the microbiomes on the outside surfaces of objects on Mars can be sterilized within a few hours on one sol. So if an astronaut goes out for 8 hours, 9 hours of an EVA on the surface of Mars, his outside or her outside of that spacesuit might be sterilized during the middle of it by the time he gets to the 4-hour mark in his EVA. So I'm not that concerned we're going to overtly ruin the surface and contaminate the surface. I think the more intriguing issue is how do we prevent bringing stuff back that might have a Martian microbiota on it, because we don't know if one exists, and if it does, we don't know the characteristics of that life. So that's back contamination. I'm not so worried about forward contamination. I'm a little bit more interested now on what might happen bringing a Martian microbiota back to Earth, if it's present, which we don't know. That's a very good
point. We're going to do Mars sample return soon, starting with the samples being cached right now from Perseverance, but very likely in the not too distant future, maybe not tomorrow, but in the next a couple of decades, once humans do go to Mars, we'll most likely bring humans back from Mars too eventually. And they'll probably bring some samples back with them. And so that possibility is really important. I do want to bring up another Twitter poll that we had through the NASA. Go ahead.
I was just going to say one other important point, though, is that absence of data does not confirm anything. So as a scientist, you have to just say, there's no data to say this. Now, don't conclude something from that lack of data. It's just no data. And so you keep your options open, you think of all the possibilities, but it is equally plausible that the surface of Mars and all the regolith is literally sterilized. Sterilized by gamma rays, sterilized by solar particle events, sterilized by all those 20 biocidal factors. That is as equally plausible as some life form that evolved 3 billion years ago actually persisting and learning how to grow, metabolize, and evolve on the surface. Equal, no data. So that's we don't jump to either one of those as a scientist. And the same thing should hold for the regulators and communities that are going to help decide the protocols for both forward and backward contamination.
Absolutely. So I will say of our audience, we did ask through Twitter, through the @NASA Astrobio Twitter account, we asked people what they thought would be the most important obstacle that Earth life might face on Mars, with options like radiation, low gravity, low temperature, the very dry conditions. And overwhelmingly, people said radiation was the largest factor they think would be an issue out of these other possible factors. So do you think, if there was Martian biota in the distant past that may still be alive or went extinct long ago, but if there were some possibility for there to be life on Mars, do you think we'd have to get a sample from deeper down under the surface to avoid the radiation to really find any signs of possible past or present life that's Martian life?
Okay, so there's two types of radiation. The radiation, the way you're using the word, refers to probably ionizing radiation. This is like X-rays, gamma rays, protons, helium atoms, etc. Very high energies, and they can ionize molecules. There's also the electromagnetic radiation of UV light. So UV light, in my view, is the strongest biocidal factor on the surface of Mars, followed by desiccation, low pressure, and low temperature. The ionizing radiation, like gamma rays, X-rays, that dosage is relatively benign from the standpoint of bacterial survival based on Earth bacterial survival under various intensities of ionizing radiation. So I think the radiation environment for inactivation on the surface is this UV radiation. The hard radiation or ionizing radiation is probably only a minor factor unless you're talking about geological time periods.
Intriguing, yeah. So that's really good to know, and a good reminder for our audience too about the processes that affect, living things on Mars, as well as possible things like organics. And so I do want to jump to our audience questions here as soon as I can,
Good.
but there are two more quick things I do want to bring up about your research that you've been working on either now or in the past. And one has to do with UV on the surface of Mars. You've been doing some work recently on studying the processes of forming and breaking down methane at the surface of Mars. And for those who are watching, they might be aware that some years ago we had the first potential detections of methane in large plumes at the Martian surface, and now more recently we've had more confirmation of methane at the surface of Mars. So what has your research been in looking at this production and destructive process when it comes to methane?
Right. So a number of years ago, I worked with several other scientists to explore how the ultraviolet irradiation environment that hits the surface of Mars is it energetic enough? Does it have enough energy per photon to break bonds in a organic compound releasing methane? And it turns out that it does. The exact mechanism for which that process occurs is not really worked out, but our research clearly demonstrated that the ultraviolet irradiation that reaches the surface can cleave or break down organics in the surface environment and in the atmosphere, releasing methane. What's intriguing is that the Curiosity rover, with an instrument called the MSL, called SAM-TLS, which is an organic detection instrument with a specific tunable laser that it can detect methane, very, very small levels of methane. It there's a number of papers published with this detection down in the parts per billion range. So 17, 15 years ago, we had data from Earth telescopes of large plumes of methane in the Martian atmosphere, followed with some skepticism by the community. Then the MSL rover Curiosity started detecting it and showing spikes in methane. And then the Trace Gas Orbiter, which is an ESA instrument or a spacecraft, it is now completely gone negative on that methane story. It cannot detect it, and it's setting extremely low levels for methane in the bulk Martian atmosphere. So what I think is happening, based on just the literature available, is that the methane concentrations that are being detected by the Mars rover Curiosity could be very close to the ground, maybe only up to a few tens of meters to a few hundreds of meters. And then those are dissipated through the day. And when the methane is diluted out in the bulk atmosphere, then the Trace Gas Orbiter is having trouble seeing that methane. Now that's an open area of study, but if we can confirm that methane exists, it has to constantly be resupplied, because the solar UV breaks down methane, which is what my research and others have shown. So it's a very interesting topic, because if it's there and we can confirm it, it could be a biogenic signature, a biosignature of some subterranean methanogenic community. It might be 3-4 kilometers down into the crust of Mars, but if it's present, those will seep out eventually, and that's what we might be seeing. But I'm starting to get into speculation here. So sticking with just the data, we have these, first there's big plumes, then a local detection, now the Trace Gas Orbiter is saying, well, not so fast, maybe it's not there.
Yeah, it's rather incredible. And your career, you've done so many different things, and I love that about astrobiology. Myself, everyone I speak to in the realm of astrobiology hasn't just focused on one individual topic. We've kind of touched all these different areas. And astrobiology itself, that's kind of crucial when it comes to understanding the nature of life. And one thing you worked on in the past was this thing called DART, the Dust Atmospheric Recovery Technology, for kind of looking at plant and human pathogens in dust plumes coming from Africa, coming across the world and kind of coming here. And that really sparked my own interest a lot. I've been involved in the past with some undergraduate level research and looking at aerobiology and how organisms are lofted into the atmosphere and move around. A very good friend of mine, David Smith at NASA Ames, he did research during his PhD looking at organisms being lofted from the Gobi Desert and for 8 days making their way across the world and kind of arriving, through the atmosphere to California. And then, we've now learned that the way the Amazon rainforest is being fertilized with phosphorus is from dust from the Saharan region of Africa being lofted and coming over here. And it shows this beautiful connection of life on Earth. But then, you were also involved in this realm of kind of thinking about pathogens for plants and humans being lofted up as well. I wonder if you could speak about, that research, what intrigued you about it, and maybe kind of your vision for these things moving around the planet and this connectivity of life.
Yeah, exactly. The planet, the planet's atmosphere circulates pretty much everywhere. So if, in theory, something is present in the Saharan desert, that will eventually be recirculated all over the planet, eventually dropping or depositing out in the Arctic regions and the equatorial regions. And the amount of dust being moved from the Sahara to North America is quite dramatic. Just in Florida every year, 50 million metric tons of dust comes from Africa. usually in the months of July, middle of July through the middle of September. But over in the Northwest, this would be Northern California, Oregon, and Washington State up into British Columbia, they're getting about another 60 or 70 million metric tons of dust coming from the Chinese, the Gobi Desert in China. So there's a large volume of it, bringing a lot of nutrients. Most of the fertility, not only in the Amazon, but in the islands in the Caribbean, are due from the minerals that are coming from over geological time, have come from the Saharan desert. So this global movement is very interesting, and there's a concept called emerging pathogens. And there are a lot of institutes that study emerging pathogens. A pathogen that was not present in one area suddenly becomes very active. It could be a plant pathogen, it could be an animal pathogen, it could be a pathogen to fish or aquatic ecosystems, or it could be a human pathogen. So I just became, as a plant pathologist, I became very intrigued on whether or not plant pathogens were being moved around in these dust storms. And the evidence pretty much supports that they are, but we don't have it really kind of well characterized, what the volume of those plant pathogens are and how much impact, if any, the plant pathogens are doing to the agricultural systems in these regions.
Wow. Yeah, and so for any of our audience who are watching, who are interested in this realm of research in microbiology and plant pathogens, how plants function in general, there's so much to take from this that you can be an astrobiologist and work on so many of these different kinds of topics. I do want to move now. I know that we're running out of time, and so I want to move now to our faster than light segment. This is where we just have some rapid fire questions. And so I just have a few that I want to ask you to bring up some of these topics that are relevant to astrobiology, and a lot of us kind of want to know from all the astrobiologists we speak to. So the first question comes from Enrico Fermi when he first asked, where are they out there? What is your favorite answer to Fermi's question, where are they?
Oh, fascinating. I just read a book called Where Is Everybody? And it's about trying to solve the Drake equation, which was developed in an attempt to answer Enrico Fermi's question. And they gave 50 reasons why aliens haven't been in contact, 50 plausible hypotheses. Of all of those, I think the most reasonable is that the astronomical distance between star systems are so incredibly huge that it is fundamentally impractical for aliens to travel between star systems. And just as an example, Alpha Centauri, which is a cluster of 3 stars sort of in this really incredible dance going on, 3-dimensional orbits, that's 4.26 light years away. Current rocketry would take like 80,000 years to have a spaceship reach the closest star. So that, I think, is the best solution. The distances are just way too big to have this easily happen.
Absolutely. I mean, there are crazy vast distances in space that kind of bend the human mind. But I'm glad you mentioned a book. Our next question is what stories have inspired you to want to pursue your career and learn more about life in the universe?
Well, I got to tell you, it goes back to 4th grade. I won't tell you the whole story because it's more complicated, but fundamentally, I was struggling in 4th grade. I didn't like to read. I was bored. I was a class clown in the back, causing trouble. And we had to buy a book from a book club. So I brought in my 50 cents, plopped it down, and I was looking around. You can imagine a little kid. Oh, this is so boring. And then I came across a title that said Mission to Mars. And I said, that sounded like fun. So I bought this book. It was about a family going to Mars, and they meet Martians. The Martians help them survive because their life support system wasn't working right. And so it's really been fun now to work, and it closes the circle. I had a new spark in 4th grade to think about outer space and flying to other planets. And that came from that one single book. And now I'm working on Mars astrobiology. I think that's a really cool circle.
That is really cool. You're speaking to that young kid in me who used to go to the Scholastic Book Fair and buy a bunch of books for a nickel or a dime each, and then get nerding out over the science and then the culture and the history and all these things.
Exactly.
And now in your career, it's been quite a while since 4th grade. You've done so many cool things in your career. If you could, though, go back to the very beginning of your career and give yourself some advice, what advice would you give yourself?
Wow. Get done with your graduate work as fast as possible, but doing excellent work. Don't dwell on stuff. And get out into the academic environment if that's where you want to go, research in academia or at NASA centers or other national labs. Get your graduate work done quick, get out, and start making a name for yourself and doing some fun work. And the second advice is pick really cool field sites. So doing that dust collection system, I was able to fly in a F-104 starfighter jet, something I've wanted to do my whole life since I was just a little kid. So, just have fun but get to it.
Love it so much. Well, let's talk about the things that excite you then. When you were a kid, you were excited by this F-104 starfighter. But right now, not just in your career and the research you've done, but just in general, what excites you about the future?
Well, I am just totally blown away to be basically just amazed on the natural world. The Big Bang happened 3.8 billion years ago. We started with primarily hydrogen, and now we have everything that we have. And there is a very logical, rational process from going from hydrogen gas in the early universe all the way up to complex metazoan animals like humans and giraffes and penguins. And that whole wide range on how soil chemistry played into it, evolution of stars, there's different populations of stars, metal-rich stars, metal-poor stars, all of that comes together, and there's just this amazing universe out there. And I'm blown away by some of the documentaries that are on TV and in broadcast YouTubes and things like that. It's just wonderful.
Yeah, definitely. And I was a young kid. I would watch like the Discovery Channel and the Learning Channel back when they had actual science content, and I would get so just amazed by these documentaries and things. I remember one from the 90s called Solar Empire that just it blew my mind thinking about what we were learning about science. And that brings me to my last question in this Faster Than Light segment. What is a science fact that maybe you still find unbelievable or that still blows your mind?
That we are here. It can be stated that simply. The Big Bang, we know when the Big Bang started, 3.88 billion years ago. But what's the trigger? Where'd that energy come from? And the fact that, again, we went from hydrogen gas through stellar evolution, planetary evolution, biological evolution, and here we are, a species capable of thinking about all of that. That's what really sort of blows my mind.
That's beautiful. Yeah, it kind of blows my mind too. Just the fact that we get to exist for even a short window, each of us, in this world, this universe, this cosmos, and some of us get to explore it together and learn more about our place.
Exactly.
I really appreciate that so much from you. I'm going to open it now for our audience Q&A. I know we have a bunch of questions coming in from YouTube and the live chat. And so let's just get started and see what everyone wants to ask. First, starting off, user @renderingreality on Twitter has said friendly bacteria like strains of Bacillus subtilis used for watering or foliar feeding make organic farming a near closed loop system. Is there any research using good bacteria or bacteria from Earth on making an organic closed loop system for Martian farming?
There is actually research in that topic, both in academia as well as in NASA. These growth-promoting beneficial bacteria, fungi, prokarya, amoeba, algae, a whole diversity of microorganisms are going to be present in any lunar bioregenerative life support system or a Mars bioregenerative life support system. And we have got to understand how those beneficial organisms are going to interplay with the plants and with the humans in these closed environments. And so because humans will be in so much more intimate contact with the life support systems on Mars or on the Moon. There's more to understand on how beneficial organisms interact with our own microbiomes. So there is research underway, and it's a really exciting area of future work for the space program.
Wonderful. Our next question comes from Roshan Bukhari on YouTube. Roshan has basically asked, what role do you think AI will play when it comes to future robotic explorers looking for life on Mars?
Oh, AI. Well, I'm worried about AI because I saw this documentary called Terminator, and just kidding, just kidding. No, AI is a very important scientific area of study, and it's so, I don't know how to predict how it's going to really play out. So I almost have to say I had no comment on this simply because it could be so amazing, and then yet maybe it doesn't really reach the full potential that we think it might be able to reach. So I think advanced computing is absolutely key to successful missions to Mars and to the surface of the Moon. And every time I struggle to get my little iPhone to work or my TV programmed, I think, golly, we're going to have trouble going to Mars, but AI is going to help us solve that. That's about all I can say, really.
Yeah, absolutely. There's a lot of advanced technologies, not just around the corner or coming up tomorrow. They're here now. We are using a lot of advanced technologies. And so our next question from user Tessellated on YouTube is about not an advanced technology necessarily, but one about using tissue culture and whether we can apply tissue culture to astrobotany. I think for myself, first, I will say, I've talked to people out there who are, in the process of developing companies to do things like lab-grown meat, lab-grown fish meat, for instance, making sushi-grade tuna in the laboratory or making beef hamburgers in the laboratory. I do think eventually we're going to see people who are learning now how to do tissue culture will be going out with their friends who are organic chemists and things like this and learning how to develop lab-grown meats for the space station and Mars. But Dr. Schuerger, what do you think of using tissue culture in astrobotany?
Well, there are many different types of tissue culture, which you just mentioned. There's tissue cultures that produce pathogen-free plantlets that would be then propagated into your bioregenerative life support system. I think that's a done deal. That will be used. There's tissue culture to create perhaps genetically modified crops that have vitamins added in that might not normally be there. One of the big concerns with humans going to Mars is a long period of time. Are we going to be able to preserve vitamins? In the normal little pills, they begin to degrade over reasonably short periods of time. So we might need to grow plants that have vitamin C in them, that are genetically engineered for vitamin C. And tissue culture has a role to play in that. Having the astronauts eat artificial fish and artificial meats, it's very, very plausible scenario. Although I'd really hate not having a nice ribeye, bone-in ribeye cooked over the Barbie. So, but the technology will be a part of these space exploration efforts.
Absolutely. Our next question comes from user Haley Monaco on YouTube, and it's an interesting one. How could we differentiate between geological methane and biologically produced methane on Mars? Is there a way for us, given current technology, to differentiate?
Yeah, we can do that now on Earth, and it's really due to the isotopic fractionation, it's called. Carbon-12 is the primary carbon atom that makes all of us. There's a small amount of carbon-13, which has one extra neutron, and then even a smaller amount of carbon-14. And biological methane has a different carbon-12 to 13 to 14 ratio than abiogenic methane. Generally speaking, abiogenic methane will not be enriched in carbon-13 or carbon-14. It'll be sort of a straight ratio of, I think it's 98% carbon-12, 1.9% carbon-13, and then 0.1% carbon-14. But biology has the ability to enrich some of those isotopes. And so that's one way to tell the difference between biogenic methane and abiogenic methane.
Yeah, absolutely. And that's important. It's also important to know the source of the carbon isotope reservoirs that we might have on Mars. And so that will require a lot of work too, figuring out, what are we starting with for isotopic abundances on Mars when we have a sample that methane's coming from. Now we do have a user, Mark Lumsden on Twitter, has asked a question: if we have any evidence of life possibly being based on different chemistry. People often ask, like carbon-based life versus silicon-based life. Could there be other solvents out there besides water? I will personally say, in answer to Mark, silicon-based life is a possibility. It might be something that we actually create. Maybe post-biological life on Earth will be silicon-based because we've made it that way. It is worth looking into the chemistry, though. Silicon-silicon bonds are not nearly as strong as carbon-carbon bonds, and they're also not super stable in water like carbon is. And so there might be a different environment. Aprotic cryosolvents, for instance, are a place where silicon-silicon bonds are much stronger and can survive a lot better. And so it's worth looking into the chemistry if you're really interested in this question, Mark. However, Andrew, for you, our next user, THM on YouTube, has said that they're interested in researching microbes living in lava tubes. Do you have any recommendations for them of what they might want to look into when it comes to understanding how microbes would thrive in a unique environment like that?
Wow, that's a very good topic. I sometimes think back on a discipline that I wish I would have gone into from a career standpoint, and cave microbiology is one of those topics. I've been into some caves in the southwest of the United States that on the outside of the cave, it's dry, hot, desert terrain, desert ecosystems. You drop down into the cave, and there's literally fog in the cave. Just a few tens of meters from the cave opening, there's literally fog forming, condensation on the walls with an incredible diversity of microbial species on the walls and on the ceilings. When I first saw that, 10 years ago on a field trip at a conference, it was like, wow, that's so exciting. How to get into it? The best thing to do on any discipline of how to get into it is look into the literature on who's publishing in the last few years on your topic. So if you find papers of a scientist that's published, on the southwest lava tube caves in a variety of locations, contact them. If their literature is not in your topical area, but it's in cave microbiology, but what they're doing is not that interesting, contact them and say, this is what I'm interested in doing. Who would you might recommend that would be a different professor? And so the best thing is just get into it, find out what you're interested in, and write to the people and ask for a little bit of advice. They mostly will respond to you.
That's fantastic and great advice. It's one way to find really good mentors and advisors out there is just to reach out to people who are doing the work. But it's also a great way to make those connections to find people who can help you find the research. And so I really appreciate that as well. It's always advice that I love to hear given to people. Our next question comes from Astativa Rai on YouTube. And I'm gonna change their question a little bit. They want to know how important is the criterion of salinity in the Martian soil when it comes to living things, including growing. And so for the Martian, and we call it regolith, for Martian regolith, not just salinity, but how important are the actual salts themselves? For instance, I know an astrobotanist who has pointed out that in the Martian, that Mark Watney eating those potatoes would have been in trouble because he didn't clean the perchlorate salts out of the Martian regolith first. So I wonder, Dr. Schuerger, if you can speak to the Martian regolith, its chemistry, and maybe how important some of these salts are.
Right. Boy, that could be a whole topic, an hour-long topic just on the geochemistry of the Martian regolith. The main thing is that the salts on Mars, the regolith is composed of ground up basaltic material that's been weathered or acted upon over geological times with water vapor, and other energy sources like UV radiation, with water vapor, with mixing of minerals. So there is actual weathering of the soils, much more than on the moon. And here's a great example. I got to use my little show and tell here. In this hand is a gray lunar simulant that I use in my lab. This is a primary mineral. It's just like ground up basalt, which is like lava. Okay, this is the Martian simulant, and it is orange-like. Now, the chemistries of these two are very similar. They're base chemistry. This is basaltic. This is basaltic. But what's happened here with the Martian simulant is that water and oxygen have acted upon this material over geological time and have altered it such that it's more complex in its geochemistry. It has a lot more salts in it. The salts are forming and dissolving and forming and changing because of this liquid water environment that's been on Mars in the past. So these two little soil samples are very different, but yet they started out at the same, which is basalt. And so on Mars, there is a problem with some of these soils might have salts that are too high a concentration to use those soils immediately for growing plants in. So we might have to figure out methods of leaching out the salts or chemically extracting the salts from the soils before they can be used to grow plants on. So that's one thing. The other thing is that the salts themselves, the little tiny microcrystals of salts, can land on a spacecraft surface, and if they come into contact with a bacterial cell, that little supercrystal of salt can actually kill a lot of different bacterial cells just by contacting. You don't even need liquid water to have the salt dissolve in. So salts are one of those biocidal factors that are in the Martian regolith. They can kill bacteria on spacecraft surfaces, and we might have to learn how to remove the salts before we can use the regolith for growth. And that's actually a really important area of research that needs to be done.
Yeah, absolutely. It's so important for us to know how we could use that regolith to grow and to use as a building material as well, to reinforce our habitats, to things like that, to build habitats. There's so much to learn. Our next question for you comes from Milton Mendoza Jr. At MS Mendoza Jr. on Twitter. They want to know that if there is life on Mars, Martian life, not Earth life, but it's not based on things like DNA and proteins, would there still be a way for us to understand biological things like how they're resisting radiation, salinity, maybe even if they're there?
Well, I think the scientific community believes or uses the definition of life that requires a series of steps that leads to evolution, adaptation. So life has to be able to take in whatever liquid they're using as their medium. In our case, it's water. Life has to take in nutrients. Life has to have the mechanisms within the cell or cells to take those raw materials and make whatever they need to make in order to divide and then keep the process going through multiple cycles. And that process requires a roadmap, a genetic roadmap that could be DNA. Well, in our case, it's DNA, but it could also be DNA on the surface of Mars, because our two planets have exchanged rock debris early in our history and continue to this day. So if life was present on early Earth, it could have been transported to Mars, or vice versa. So life requires this process, and at the end of that process is evolution. So if there is an extant microbiota on the surface of Mars or in the shallow subsurface, it has to have those ingredients. It has to be able to acquire nutrients, which means we should be able to figure out what it needs as a set of nutrients. It's going to be sensitive to some things and not to others, and we should be able to figure that out. The real question is, on Earth, greater than 99% of the microorganisms that are in our environment we're not able to grow them in the laboratory. So that creates quite a lot of difficulty in looking at their basic metabolism, basic methods of evolution and adaptation. There are methods to doing that, and I think those same methods can be applied to any microbes on Mars that are not culturable. But the last point of this question is that, like Earth, Mars, if there's life, has a series of ecosystems. It's not just a single bacterium sitting on a rock saying, hey, I hope I get sunlight today. there's an ecosystem there, and there has to be energy flow within that ecosystem, and it has to be chemical flow and disequilibriums, creating the ability to do work in that ecosystem. That is something that's really complex, and it's like we should be able to see that. If there's life, then there's ecosystems, and we should be able to detect those, I believe, without invoking very unusual, exotic silicon-based life or methane-based life at minus 300 degrees or whatever.
That's awesome. So I so much appreciate your time in joining us here. I apologize.
It's been great. Thanks.
So many more questions, but we are at the very top of the hour. If I could just ask for a really brief answer to one more question. I saw one that I really liked from Kashish Gupta on YouTube. So Kashish has asked Dr. Schuerger, if you get to send a single microbial species on a future mission to Mars, which one would you pick?
Wow, I don't want to send any microbes to Mars. I would pick a cyanobacterium capable of starting the process of terraforming the planet. If we understood the environment on Mars enough, and humanity is ready to take the step towards terraforming the surface of Mars, my first thought would be to begin sending microbes that can take carbon dioxide out of the air, produce oxygen, maybe even utilize the nitrogen. So that would be, I think, the most intriguing. But that's tens of years away, if not hundreds of years away.
Absolutely. Yeah. Well, thank you so much for that answer. To the rest of our audience, I apologize we couldn't get to all of your questions. There were so many good ones there. If you are interested, you can always ask your questions in the comments section once we post the recorded version of the YouTube video as well. For those of you out there who watch the show and join in and ask us questions, please share the show with your friends. We love to always point out an ambassador of the month for each episode who's shared about the show, who's asked questions of our guests, who's really gotten engaged in this whole thing of astrobiology. We want to give a special shout out this month, for instance, to Fathi Karuia. Fathi is an affiliate researcher at Blue Marble Space Institute of Science, as well as being a researcher at NASA Ames, and he shared about our show. So Fathi, we really appreciate that. Dr. Schuerger, thank you so much for joining us for Ask an Astrobiologist.
Oh, hey, this has been fun. This has been fun. Thanks.
Yeah. Thank you so much. I'm glad our audience asked so many great questions about your research. They might even reach out with more questions, so keep an eye out for that. For those of you tuning in and joining us, I hope you signed up already for the NASA Astrobiology mailing list. That way you can find out about new episodes of Ask an Astrobiologist, as well as all of the other incredible things going on through NASA Astrobiology, including opportunities for early career researchers, missions, new articles on the website, lots of cool things going on there. So please sign up for the official mailing list. Thank you, Dr. Schuerger, for joining us. Thank you to the audience for joining us. And as always, everyone, remember to stay curious. Bye, all.